Optical methods such as optogenetics can provide the precise modulation of cellular molecular signals. Our protocol demonstrates an all-optical manipulation of cellular signal moleculars by femtosecond laser stimulation. By coupling a femtosecond laser into a confocal microscope, our technique can provide single-cell or subcellular level operation by femtosecond laser stimulation and real-time molecular dynamic monitoring by confocal microscopy.
This method can be applied to any fluorescent microscopy systems by coupling a femtosecond laser into the system in a proper manner. For anyone who is trying our method for the first time, please follow the safety instructions of the laser facility or seek guidance from professionals when operating a femtosecond laser source.
To begin, turn on the femtosecond laser and the confocal microscope. Use reflective mirrors to direct the femtosecond laser beam through a mechanical shutter. Next, set a relay telescope consisting of a pair of lenses to expand the transmission beam width. This should be consistent with the back aperture of the objective. Now, steer the reflective mirrors to align the expanded beam into the microscope. Adjust the two reflective mirrors pointed out here to tune the focus of the femtosecond laser to the center of the field of view. Finally, measure and tune the lasers as described in the accompanying text protocol. Culture HeLa cells using standard techniques. When ready to perform the experiment, transfect the cells with ERK.